Welcome to iNovaScience, where curiosity sparks discovery. Subscribe and join the journey.
What if we could design a super protein, one that doesn't fall apart, even when boiled? Scientists at Scripps Research may have just done that. They've discovered a hidden atomic pattern that makes some of nature's most fragile proteins incredibly stable, and it could transform how we design new drugs and biotechnology. So, how did they do it? Let's dive into the microscopic world of cell membranes where billions of proteins are constantly working to keep us alive. Every cell in your body is surrounded by a thin, protective layer called a membrane, a kind of molecular skin made from fat-like molecules called lipids. But this barrier isn't just a wall, it's alive with motion and control. Tiny molecular machines called membrane proteins sit inside it, doing essential jobs, moving nutrients in, sending signals out, or helping cells stick together. These proteins are like gates, antennas, and glue, all rolled into one. When they malfunction, diseases like cancer can arise. That's why scientists spend billions every year studying and targeting them. But here's the problem. Studying these proteins is hard. As soon as researchers take them out of the membrane to look at them, they fall apart like trying to lift soap bubbles with your hands. To truly understand how these proteins work, scientists need to see how their atoms hold together. But natural proteins are fragile and unpredictable. So the team at Scripps Research came up with a bold idea. What if we designed our own proteins from scratch using computers? Instead of struggling with nature's complicated versions, they could create synthetic proteins that mimic the real thing but are stable enough to study. Inside these proteins, the researchers noticed something strange. Many of them shared a repeating pattern, a small amino acid every 7 positions in their sequence, like a rhythm hidden in the code. This pattern appeared on the same side of every spiral or helix in the protein. They suspected that these repeating sticky spots help the helices grip each other, giving the whole protein its shape and strength. To test their idea, the team used a computer program to design new proteins built around this pattern known as the Gly-X6-Gly motif. Then they made these proteins in the lab, and what they found was astonishing. When the scientists produced their designer proteins, they didn't just stay stable, they stayed folded perfectly, even under boiling conditions. That's right, proteins that don't fall apart when boiled. The secret? It turns out that these motifs create a special kind of hydrogen bond, a weak attraction between atoms that becomes incredibly strong when repeated over and over. Individually, these bonds are fragile, but together, they form a powerful network, a molecular handshake repeated hundreds of times. Nature, it seems, has evolved to use this subtle pattern again and again across life. Now that we understand how this motif works, scientists can use this knowledge to identify genetic mutations that might break these bonds, possibly revealing why some diseases occur. Even more exciting, researchers can now use their computer program to design new membrane proteins or even create molecules that target them directly in the body. This discovery could speed up drug design, bioengineering, and even the development of new therapeutics that work precisely where they're needed, inside the cell membrane itself. In a world where biology and technology are becoming one, this study shows what's possible when we learn nature's secret codes. By understanding how proteins stay strong, we may soon design life's building blocks with the precision of an engineer and the creativity of evolution itself.
Subscribe to iNovaScience. See you in the next video.